Good evening, everyone. My name is Vivian Manso, and today we're going to talk about the correlation or the relationship between the human gut microbiota and diet-induced obesity. And what got me interested in this particular topic is microbiology. And in my presentation, I would like to talk about how these two are correlated to each other. Before I start with my presentation, I'd like to give you an outline of how I'm going to present this information to you all. First, I would like to give a background information or knowledge about human gut microbiota and diet-induced obesity. Then, I would like to talk about the connection between the human gut microbiota and obesity. Then I'm going to be talking about experimental studies on how these two are being correlated using exercise and a legume called MUNG. Then I'm going to talk about how this is relevant to the human population in the world. And I'd like to leave with you all with a take-home message. And finally is my reference page or slide. First, I'd like to talk about what human gut microbiota is. The human gut microbiota is a microbial composition in the gut. Basically, it's the gut ecosystem of all viruses, fungi, bacteria, and other microbes in the gut. These microbes help with the digestion of fats, fibers, and other food in our body system. It helps with metabolic processes in the body system as well. And we need these bacteria or these microbes to help with the body function. These microbes are good microbes. They do not cause any diseases, they do not harm the body or cause any infections to the body system. In the human gut microbiota, we have good bacteria and we also have bad bacteria which causes infection. But in this presentation, I would like to talk about bacteria only. And in the gut system, we do have a phyla in the gut system. So we do have a lot of bacterial phyla in our gut system. And in this presentation, I would like to focus on two specific phyla, which are the Phyla Firmicutes and the Phyla Bacteroidetes. And we have other phyla present like Actinobacteria and Proteobacteria. Now, I would like to talk about what diet-induced obesity is. Diet-induced obesity is caused by an intake of high-fat diets or a diet rich in calories. When one is obese, it puts one at a risk of getting diabetes, cancer, cardiovascular diseases, heart-related diseases, and other lipid metabolic disorders. This obesity is prevalent in adult population. I also like to talk about how socioeconomic factors could also play a role in diet-induced obesity. So, to talk about the role of socioeconomic factors, not everyone has the same money, or not everyone has the same financial status. We all have the rich, the poor, the middle class, and all status. You know, when one is rich, one is able to get good food, organic food, and other food substances that could be really helpful to the body system. Unlike the poor, or unlike somebody who doesn't have enough money, who settle down to something that they could settle down to something that they could afford, and which could be something like probably not organic wise, but something they could afford to order to feed their family. So we can see that there's actually socioeconomic factors playing a role in terms of who gets the best food and who doesn't get the best food. And that really play a really major role. And I think it's really important to understand that obesity could also be as a result of societal or social implication. Now, I would like to talk about how these two terms are connected to each other. So, as your normal gut microbiota, you want your Bacteroidetes to be higher than the Firmicutes. And anytime you're taking a high-fat diet, this changes. So now you have an increase in Firmicutes and a decrease in Bacteroidetes. And why does this change? So the Firmicutes is responsible for absorbing fats. So as you take in more fat food, there's an increase in Firmicutes. And while there's an increase in Firmicutes, this adds on to the body weight of the mice, and this leads to obesity or overweight. So an increase in Firmicutes could lead to an imbalance in the gut microbiota. So diet is definitely a factor that plays a role in changing the gut's microbiota, which could lead to one getting diet-induced obesity. Another factor could be age. So as you age or as you grow older, you could see that the diversity of your gut microbiota lowers, or you could see a decrease in the diversity of the microbiota, and which could put one at risk of getting obesity. Second factor, or the third factor, could be environmental factors. So there was a study that was being done where they compared people from Africa to people to people who took in African diet and to people who took in Western diet. And you can see that these two population or these two people had different microbial composition because of where they come from. And this could be narrowed down to their diet. So environmental could also play a role in diet-induced obesity. In this experiment, I'll walk you through two experimental studies that was done to help in creating that shift or help to diversify the microbial population. In this case, help to bring back the imbalanced gut microbiota to a balanced gut microbiota, which is more Bacteroidetes to less Firmicutes. One thing that I would like to add to this is that there is little research or knowledge done in this field of study. More research is being done in order to find a therapeutic target of how we can combat diet-induced obesity. Now, our first experiment is going to be about mung bean. So this is what a mung bean looks like. So mung bean can be used in stir fry, could be used in our soup, could be used as an ingredient in any type of food, you would like it to be. This mung bean could also be used in China as a medicinal component. This mung bean is really essential and very helpful. It has a lot of health benefits. It has anti-cancer benefits. It has hepatoprotective ingredient, which is it helps your liver, and other benefit that could help one from getting chronic illness. This mung bean is also a photochemical or antioxidant, which kind of help the body from any oxidative damage and also help one be at a low risk of chronic illness. So in this experiment, they used a mung bean to see if this mung bean could actually cause a shift or actually restore the balanced gut microbiota. So what they did was they used mice as the model. And the reason why they use mice as a model is because mice tend to have the same microbial composition as humans. And that's why they use a model. A model is supposed to represent something that you're studying. So they use a mice model, especially for diet-induced obesity. So in this experiment, they categorized mice into 3 groups. So in the first group, as you can see, let me see if I can find my arrow, mouse pointer. Okay, got it. So in first group, we have the NCD group, which is the normal chow diet. And that could be a regular cat food or kind of like a green diet or a cereal diet. And then we have the HFD, which is the high fat diet. And then we have a third group called the HFD CMB, which is a high fat diet plus a mung bean. So in all, we have 3 categories of mice. We have the normal chow diet, the high fat diet mice, and then the high fat diet with mung bean mice. So they did this experiment over 12 weeks, and what they did was they took the fecal samples and they extracted the bacterial DNA and isolated that, and the PCR using specific primers to encoding genes of the bacteria present in the gut system of the mice. The purpose of this figure is to show the major, and let me move this, is to show the major Phyla present in the gut system of the mice. So I know, as you can see more green and also more red. Really, you can see blue and purple, but they are not as much. And as you can see, there are more green and red, as I already said. This green corresponds to Firmicutes, and then the red corresponds to Bacteroidetes. That is why in our previous slides, I did say in this presentation, I'd like to concentrate on two phyla, which is the Firmicutes and Bacteroidetes, because these two phyla play a major role when it comes to diet-induced obesity. So after they did the sequencing and the PCR and did the encoding genes for Firmicutes and Bacteroidetes, they had these results. So in results B, you can see that NCD, as I already said, which is the blue, is a normal child diet. You have your red being your high-fat diet, and you have your green being your high-fat diet with MUNG bean. So in this figure B, I want us to just focus on Firmicutes and Bacteroidetes. So for Firmicutes, we have the blue, which is the normal child diet. And as you can see, the Firmicutes is relatively low, but when you take in a high-fat diet in red, you can see it increases the Firmicutes, and that's not what we want. In a normal gut system, we want it to be very low as the normal child diet. Now, the green, which I'm pointing, is the one plus the MUNG bean. You can see it changes it a little bit, like it kind of reduces it, but not as much. But there's still some kind of decrease. You can see the MUNG bean is trying to restore that Firmicutes by making it lower as compared to the high-fat diet. Now, let's go on to the Bacteroidetes. So for the Bacteroidetes, we have blue, which is the normal child diet. You can see there's an increase in the Bacteroidetes, and that's what we want. We want our Bacteroidetes to be high. But when you take in high fat, you can see the population of the Bacteroidetes reduces. But an interesting thing happens here when you add high-fat diets with the MUNG bean. You can see it actually increases and actually wants to restore the number of Bacteroidetes in the gut system. So we can see that MUNG bean is helping create a shift is helping to return the gut system to its normal balanced environment. For C, I know you all have taken biostatistics from Dr. Donnelly or other science professors that teach biostatistics. And I know you all know what this is. This is a box plot. And on my y-axis, I have the F over B, and that is Firmicutes over Bacteroidetes ratio. So as you can see in the blue, which is the normal child diet, we have a low F over B ratio, and that's what is expected. We expect our Firmicutes to be lower than our Bacteroidetes. But when we take in high-fat diets, which is the red that we can see, you can see that it's really high. So you have the middle line being your median, and you have these lines being your interquartiles. So you can see that there's a high ratio due to the high fat, because the high fat increases the Firmicutes because the Firmicutes absorb the fats. So therefore, as you take in more fats, you're increasing the Firmicutes, and that add on to the body weight. But what is really very interesting and very surprising is when you look at the green, which is the high-fat diet with the MUNG bean, you can see that it dramatically lowers the F over B ratio, which is the Firmicutes over Bacteroidetes ratio. And according to the experimental study, it actually was very significant. That MAC actually significantly created a shift in the environment of the microbiota, and actually helped restored the number of Bacteroidetes in the gut. And this is really helpful because now changes in our diet could actually create a balance or an imbalance in our gut system depending on what kind of diet you take in. And we know that the mung bean actually helps one who is probably obese, which is the mice, to kind of restore its microbiota to a balanced one. This second experiment, as you can see, was the mice performing an exercise, which is really interesting. So in this experimental study, they were also trying to see if exercise, which is a lifestyle change or something that could, or a practice that could create a shift in the gut microbiota of an obese mice. So as you can see, this mice is being put on a wheel, and the movement of this mice on the wheel serves as an exercise or as a practice for the mice. And this is how they measure the distance using an instrument called odometer. So in this experiment, they used a mice model, and they had 4 categories of these mice. So the first category, they had a mice that took in a low-fat diet, and the second category, they had a mice that took in a low-fat diet and exercised. The third one, they used a mice that took in high fats but never did anything. The fourth mice group took in high fats and performed exercise. So in this figure is showing the distance and the distance that they did, which is the exercise on the wheel movement over the 12-week period. Now, I want us to concentrate on B. So the red is high fat SED. So SED means they didn't really do any exercise. It's like sedentary. They didn't really do anything. So as you can see in this diagram, once again, where did my mouse pointer go? So right here, there we go. So we have our high fat SED, and as you can see, the body weight increases because it doesn't really do anything. All it does is taking high fat and doesn't do anything. But let's compare that to high fat exercise. So as you can see, the high fat exercise, which is the green line, there's a decrease in body weight as compared to the blue line. So you can see exercise is actually causing a change in the body weight of these mice. Now, what it did was they took a fecal samples of this, they took a fecal samples of these mice. They isolated the bacterial DNA, PCR, sequenced the genes, sequenced the cysteine-S RNA genes by using specific primers to encode the genes of Bacteroidetes and Firmicutes. So here, I want us to concentrate on Bacteroidetes and Firmicutes, and I want us to really concentrate on the blue and green bars. So let's see. So figure A is Bacteroidetes. So as you can see in figure A, we can see the blue, which is really low, which is a high fat exercise, a high fat mice, which doesn't do anything. In the green one, you can see there's an increase, which shows that the high fat is actually performing exercise, and the exercise is actually creating a change in the Bacteroidetes population, because now it's actually causing an increase in the Bacteroidetes. But B is the Firmicutes. As you go to the Firmicutes, you can see that in the high fat cell, there's really an increase in Firmicutes because of the diets that they take in. But when you look at the green one, which is a high fat exercise, you can see that tends to decrease the Firmicutes. And in this experimental study, exercise was really significant to the changes in um, the gut microbiota. So, in these two experimental studies, we can see how important it is to actually how we can create a change in our diet and how that can actually create a change in the gut microbiota system in the mice. Now, why is this really relevant? So, I personally think this study is really relevant because diet-induced obesity is becoming a very significant problem in the country or in the United States of America. And it's really important to understand that the diet we take in and whatever we take into our body can actually cause damage or harm to us. So it's very important for us to know what we eat, the kind of food we take in, how much high fats do we really want to take in a day. And that's really important to consider as we go on our daily lives. Also, practicing a really good lifestyle could actually help with your microbial diversity. And this goes into exercise. So if you actually perform an exercise, I know not all of us could have access to gym. Not all of us could have access to, going around walking our neighborhood, running and all that. But I think if we practice a good lifestyle, which could be eating a low-fat diet, something to kind of keep you engage, which is probably like walking around your house, doing something that just makes you have that physical health in will be really important. And that could help with the microbial diversity in your gut. Also, this is really relevant because now health organizations in the countries are really trying to see if there could be a change in our dietary patterns, which is maybe we need to include more plant-based diets in our food. And they're actually kind of suggesting maybe a fiber or some kind of plant diet, which is like the mung bean, which could actually help diversify the gut microbiota in the mice or in humans. Now, socioeconomic factors, we cannot run away from socioeconomic factors because not all of us could afford, you know, eat good food, eat good there, go to the gym. Not all of us could afford that. So I think as scientists or researchers or other organizations are bringing up solutions, I think it's really important to also to make sure that these solutions or these diets is also accessible and affordable to everyone, regardless of your background. Now, my take home messages. So, from this experiment or from this my presentation, I'd like you to know that changes in diet could actually either create a balance or imbalance in the microbial community in your gut. And your microbial community comprises of good viruses, good bacteria, good fungi, which helps with digestion of food in your gut. Also, anytime you have any imbalance in your gut bacteria, in this case, if you have more Firmicutes than the Bacteroidetes, this could lead to one getting diet-induced obesity. Lastly, that is not the end. There are other lifestyle changes that you could do to help diversify that microbial community in your gut. And that lifestyle changes could be a change in your diet, which is eating more plant-based diets, eating more fibers, or eating more mung bean, if you like to, and to create that change or kind of restore that normal gut microbiota in the system. Also, exercise. Any form of exercise could also help create a change in the gut microbiota in the system. And as I already said, research is still going on in this particular field of study. Even though there is a correlation between gut microbiota and diet-induced obesity, researchers or scientists are trying to really understand what really causes this change, how exercise and movement actually help to restore this. It's still in progress, and hopefully they're trying to use this correlation to bring a therapeutic target of how they could actually reduce diet-induced obesity in the country or in the society. And this is the end of my presentation. I hope you enjoyed it and have a rest of your good evening.